Hi, my name is Grae Patterson, and I'm a sophomore in CBE. This year, I had the opportunity to work in Dr. Krebs' lab on the fabrication and characterization of biocompatible hydrogels for trabecular meshwork cell cultures. For a little bit of background information on my project, glaucoma is the leading cause of irreversible blindness and affects nearly 70 million individuals. It is caused by a buildup of aqueous humor within the eye, which raises the intraocular pressure. The intraocular pressure is regulated by the trabecular meshwork, which you can see in the photo. Currently, there is no cure for glaucoma, but there are drugs to treat its symptoms. Testing these drugs is difficult due to the lack of similarity between cells in culture and the native tissue. In order to conduct better studies, we need to find a way to more accurately mimic the trabecular meshwork environment. One of the ways that we did this is by using methacrylated gelatin, or GeIMA, which is known to be biocompatible and similar to collagen-based tissue. The overall goal of this project is to create an in vitro model that can more accurately model the HTM to one day be used in therapeutic studies. In order to do so, we needed to determine which concentration of GeIMA would support proliferation of the TM cells. Additionally, we wanted to test the effects of including GAGs that are present in the trabecular meshwork, HA and CS, to see if this would have an effect on the behavior of the cells. In order to determine if the cells were behaving the way they would in the body, we used confocal imaging to visualize the expression of the HTM protein fibronectin. The data shown here demonstrates that the presence of GeIMA supports TM cell proliferation for 2 weeks. However, the 3% concentration was ruled out since this gel fell apart before day 14. We ultimately decided to use 5% for subsequent scaffolds since this is the concentration recommended by previous research. The graph on the right shows the effects of including GAGs in the cellular scaffolds. Based on this data, all of the scaffold types achieved similar proliferation except for the scaffolds containing CS. Here are the confocal images used to visualize fibronectin, which is the red net-like structure in these images. Here you can see that the inclusion of CS increased fibronectin expression, while HA decreased it. Based on the results of this project, I believe that the next steps would be to try to increase cell proliferation on scaffolds containing CS, since this is the GAG that increased fibronectin expression. Additionally, the effects of adding other GAGs present in the trabecular meshwork could be tested. Lastly, increasing the structural complexity of the scaffolds to allow the cells to culture in a 3D environment may help to better replicate the native tissue. This is something that we worked on at the beginning of the semester using a 3D printer. The photos on the right are of prints we did using GeIMA as the printing material, but we believe that a 3D printer that is able to print in finer detail is needed to create the porous structures we were hoping for. Thank you to my mentors, Dr. Melissa Krebs, Dr. Matt Osmond, and Ben Stinson for helping me with this project, and thank you for listening.